Well, thank you very much for the invitation to present my work today. So, as mentioned, I moved from the UK to the Canary Islands about 10 years ago, and when I moved, I thought it was a great opportunity to shift from sort of fairly what you can call superficial phylogenetic analyses or single species studies, and try and take a community-based approach to understand what processes really are important in structuring biodiversity across these insular landscapes. Okay. Let's see. Let's be going forward for some reason. Next slide. Okay, it's working good. Right. So one of the big challenges then is that, you know, if you go and work with arthropod communities, you're generally confronted with like quite a large amount of diversity that you have to deal with. And what you want to be able to do is assign everything that you come across to a species. And, you know, that's the first big challenge, really. It's if you want to sample a lot of areas for a lot of species, you know, you have to get over this hurdle of how you're going to efficiently classify things. So what we did was take an existing ecological sampling protocol developed by Paulo Borges in the Azores, and we added a molecular component to it, another molecular layer. Okay, so it really is like a validation step to, step to validate species hypotheses. So we basically go through sort material to what we think are species based on visual inspection. We can call them parataxonomic units. Then subsample those and sequence them for a mitochondrial gene to really test for the congruence of the mitochondrial and the species level hypotheses we're making. So it has a lot of advantages. It reduces the taxonomic load. You don't have to identify everything to species, just a few individuals from your end of the protocol species that you defined. It increases the comparability among independent studies because you're working with sequences and they're directly comparable as opposed to, you know, morphological hypotheses. And it reveals a lot of cryptic diversity, which is particularly relevant in insular communities. And what is particularly useful is that this data can itself provide information about species-specific dispersal limitations. Let's see if we can hide this over here. Right. So what do I mean by this? Well, here we go. So let's just take these two hypothetical examples. Species sampled from the same 10 sites across this peninsula, which is actually the Anaga Peninsula of Tenerife. What we would do is for each of these species, we would sequence 4 individuals from each of the plot where it was sampled and generate a mitochondrial sequence, just a simple CO1 sequence. Now, what can result from this? These are the extremes that we might find. But within each species, in this hypothetical case, we've got 5 different genetic variants. And in the first one, now all variation is everywhere. This is kind of what you'd expect for a species that's not dispersal limited. Everything gets everywhere. You know, there's random mating across the range of the species. In this other example, we have 5 genetic variants, but you can see here that they're highly structured geographically. So this automatically tells us that, you know, there is some dispersal constraint within this species, some historical limitations to movement. And further to this, we can use the phylogenetic information within these sequences to understand the historical context of this dispersal limitation. So here we can group things at different hierarchical levels based on phylogenetic relatedness. In this example, we see that, well, there's 3 principal clades, and there's more recent divergence between the yellow and blue clades, which are more ancestrally connected to the darker, the black and white clade. Okay, so this information itself, within a single species, you always have to treat mitochondrial variation with some level of skepticism. But if we're working across a large number of species, it can become a powerful tool to find repeated pattern across species, which invokes common process. So what I'm going to do today is to talk to or present to you some of the earliest sampling we did in this project when we started about 10 years ago. And it involves these 10 50 by 50 meter forest plots in the laurel forest of the Anaga Peninsula on the island of Tenerife. Now, this is essentially cloud forest. It's forest that is maintained by the trade wind inversion, generating a cloud layer that this forest sits within. So we have these 50 plots, and we had a replicated sampling protocol involving 27 hours of varied sampling techniques across each plot. And the focal groups were beetles and spiders. And the fundamental data you get from this is information about presence and absence and relative abundance across plots, and microhabitat information that comes from the sampling of individuals themselves, depending on which sampling types they are captured by. So here, everything that I'm going to talk about now, you can find in more detail within this paper here. And I'm going to present the results of the beetle work from these 10 plots. So we sampled in total just over 23,000 individual coleoptera, and these resolved into 154 presumed biological species based on our molecular protocol. Now, there was another 30 species that never sequenced, so there's actually more species in the community, but there's 154 for which we obtained sequence data and for which we have fairly robust estimates of species identity. Now, 146 were native or endemic, which automatically tells us that this community itself is not terribly invaded in terms of its beetle community. And of those 146, 105 were sampled with enough individuals from enough localities to undertake some population genetic analyses. And I'm going to present now some summary results from a few species that just represent patterns that were fairly recurrent across different species. So here's one species, Laodes oceanica, and you can see the mitochondrial tree to the right. Okay, and you can see I've defined an orange and a blue clade, and these two clades correspond to these two geographic areas that you can see here. Hopefully you can see my arrow moving around here. Okay, this is the pattern that was highly repeated across species. So here we see it again in this species of weevil, Laparoceras subnudosus, and this yet to be described species of Cardiophorus, again two clades, clearly structured into those two regions. And here, another weevil found in the forest canopy, Acales globulipenis. Now, it might not be surprising that all of these species are flightless, so we know that they have dispersal limitations, but the differences we're seeing across that describe these two regions are not just simple isolation by distance, as we can see by these long branches. And in fact, some species offer even more insights. So this species, Acales globulipenis, the individuals I'm showing in red here, actually come from a geographically far more distant site. But even though, despite this geographic distance, the red and the blue are essentially nested one alongside the other and highly divergent from this orange clade here. And this is again a highly repeated pattern. And here I'm just going to summarize how repeated that pattern is. So in this heat map, we're showing for these 105 species for which we have sufficient mitochondrial data, the extent of phylogroup sharing among sites. And it's also summarized down here in this hierarchical clustering tree. And we can see that across all species, there's a very strong signature for high phylogeographic similarity among sites here and among sites here, with little sharing of phylogroups between these two regions. That's at the level of phylogeographic groupings. even with less informative indices of population structure, we find the same pattern repeated. So looking at NST values within species and GST values within species, the same structuring occurs and is equally significant. And even just looking at individual haplotypes across all species and the extent to which they're shared or not among sampling sites, the same pattern emerges. Where we don't see this pattern emerging is when we look at the level of species themselves. We don't find that those two regions are more internally similar to each other due to their species compositions, which tells us the kind of patterns that we're seeing here, although striking, are probably fairly recent in origin, as they haven't given rise to a species level signature for this regional differentiation. Now, what I'm showing you in this map at the top here is just the summary climate layer for that Anaga region. And what I'm showing you here is a climate layer of precipitation. So what you can see is that, you know, just eyeballing it, here's the two regions, one here and the other here. And we have this area of low precipitation between the two. And we hypothesize that the structure we see has something to do, has some climate involved explanation. And indeed, when we did a landscape resistance analysis using these, we're using 6 environmental variables or climatic variables related to water stress, plus including simple geographic distance in a null model. In terms of the phylo group structure we see here, it is clearly explained by elevation and precipitation, and not just by simple geographic distance. So this is a fairly coarse-grained analysis. You know, it's just like mitochondrial data across individual species. We know mitochondrial is not a a fairly fine-grained marker. So what I want to emphasize here is how, with this sort of broad brush community level structuring, we can now go in at a finer scale within species to really try and understand what the drivers are of this recurrent structuring process that's occurring across so many species simultaneously. So we generated, well, first we chose 16 species from a pool of 104. We chose 8 that showed strong mitochondrial structuring into these two regions, and we chose another 8 that show really no mitochondrial signature for any structuring across these two regions. And we generated RAD sequence data, and for anyone who may not be familiar with that, it's simply subsampling the genome for short molecular markers. So we get thousands of loci randomly distributed across the genome that we can use for a more powerful analysis of inter-individual relatedness and relate that back to landscape features. So what I'm summarizing here on this figure is firstly the 8 species which a priori showed strong mitochondrial structuring between these two regions. And here we have phylogenetic networks of the mitochondrial variation for each of these 8 species. And the black line I'm putting on there simply marks where the differentiation is between these two regions, which we call east and west. Now here is networks derived from the nuclear genomic data. Okay, so thousands of loci used to generate inter-individual relatedness summarized in a network. And for all of these species, it's the same structuring into east and west as we see with the mitochondria. So it's, I mean, for me, this isn't a terribly big surprise, but it's important to validate the mitochondrial data and to obtain the level of information required to really understand at the species level what individual agents are structuring the variation as we see it. So now I'm just representing the same data as a principal component analysis, again, clearly divided into 2 groups. And for each species, we generated ancestry coefficients. So in all cases, except for the last one, we find that the individuals clearly structure into 2 ancestral populations. Now in the last case here, we actually have an extra, we have 3. So there's another region implicated for this species here, but all species provide evidence for an east-west population. And the really interesting thing that comes out from this ancestry analysis is that in all but one species, we find evidence for admixed individuals. And these admixed individuals are in all cases from populations where these 2 regions almost come into contact. Okay. Now, when we undertake individual species level landscape resistance analyses, for all species, the geographic patterns of individual level relatedness are best explained by environmental or climate variables related to water stress. Simple geographic distance or a null model does not enter into the models for any of these species. Right. Now I'm just going to show you for context the results from these 8 species that showed no mitochondrial variation at all. Okay, so here's the mitochondrial networks, no structure to east and west. Okay, but for 5 of these species, the nuclear data did in fact reveal east-west structuring. Okay, the 3 that didn't, I've grayed them out, but for the 5 that did, you can see it's the same key feature in the analyses of admixture, ancestry coefficients. Individuals with mixed ancestry, in all cases, correlating to these populations where these 2 regions abut each other. So what the data is essentially telling us is that we have a model of isolation and secondary contact repeated across a large number of species. So we have, how many is this? 1, 2, 3, 4, 5, 6, 7, 8, 10 species that show exactly this structuring here, east-west, and a contact area here. We have another species that's divided to east-west but was never sampled here. So we have 11 species showing the same regional context structuring. One species that shows a similar pattern, but the admixture is shifted further towards the east. And another species that's actually structured into 3 regional populations with admixture coinciding with the 2 regions that I've just described here. So 13 out of 16 species show essentially the same pattern of regional structuring and secondary contact across an area here that is less than 4 kilometers in distance. So we looked at what the estimated ages of the onset of divergence were across these species using the mitochondrial data, and they range from 4.6 million years ago to less than 100,000 years ago. So the mitochondrial data is suggestive of a similar dynamic, but different species entering into the dynamic at different time points. Now, to understand what might explain that, you really need to understand the geology of the region and the climate history of the region and the local climate phenomena that support the rainforest there. So what I'm showing you here, this white line denotes the outline of a mega landslide that occurred about 4.5 million years ago. So this is it fell forward into the ocean that way. So on the other side of this line, there's a big chasm, basically, that's where a large amount of the peninsula fell into the ocean. So what that has given rise to is a lower elevation region here, all along this crest that's typically above 700 or 750 meters, but at this point here, it drops down to 620 meters. Okay, so there's this low elevation region in the crest. If we look at where the time point for that landslide, it occurred in a period of global climatic relative stability. What I'm showing here is how climate has oscillated through time in the last 4.5 million years. So you can see it's become increasingly variable and more extreme as we get towards the present. So the next thing we need to understand to put the pieces together here are the nature of the cloud formations that sustain the cloud forest that sustains these organisms. So the cloud forest is essentially a product of the trade wind inversion, which generates this cloud layer, which I'm just showing you now how it can vary on a daily time scale in terms of its altitude. So you can see it's shifting down slope as we get towards the end of the day. So it's a highly variable phenomena in terms of where it is and when it's present. Typically it's strongest during the summer months, but it varies on a daily and a millennial timescale, and it varies as a function of regional climate phenomena. And it will vary as a function of these longer term quaternary climate oscillations. So the upper and lower limits of the cloud layer, which define the upper and lower limits of the cloud forest, will shift through time. What we really don't know is how they shift through time. So here I'm showing you how sorry, what we know will be happening through time. Cloud shifts downslope and upslope. So when it's at its highest elevation, cloud forest will be restricted to the highest elevations of the peninsula. And as it moves downslope, cloud forest will expand downslope and isolated patches will start to coalesce. Intuitively, we expect that as climate warms, the cloud layer should be pushed upslope. Warming climate, intuitively, we understand results in distribution limits of species being pushed upslope. And that's summarized in this model I'm presenting here, the lifting cloud-based model. Okay, so in warmer climates like now during interglacials, rainforest is expected to be, cloud forest is expected to be restricted to the upper slopes. And then during glacial periods, it will be more extensive, extending to lower elevations. There is an alternative model, which is exactly the opposite, which predicts something that you might think is quite counterintuitive, that as climate warms, the cloud layer is pushed downslope. And this is all because of a complex interaction of global trends of global warming or cooling, and how such phenomena as the intertropical convergence zone and the El Nino Southern Oscillation and the Hadley circulation respond to these changes. And the evidence for this comes from a number of independent studies from different regions around the world, from the Dominicas, even from the Canary Islands itself. So what we do know already is that we have this repeated pattern across independent species involving isolation and secondary contact. And because it's repeated across so many species, there's a common driver, which can only really be due to this interaction of climate and topography. So we can now go to an even finer scale within a species to test between these two competing climate models to really understand not just what's driving divergence within these species, but how the cloud forest ecosystem itself is regulated by regional climate and how it's expected to change with climate warming. So what we did was we chose one of our species, Acales globulipenis, and sampled more fine scale with more sampling points across this dorsal ridge cloud forest. Now I'm just going to show you that in a different orientation now. We'll flip this around. So we're looking from west to the east, and we generated RAD sequence data for this greater sampling of individuals. Okay, and I'm just going to summarize the results for that now simply. So here I'm showing you individual ancestry coefficients for individuals sampled from each of these populations. There's 19 populations, and you can see I've grouped them here by their numbers, 1 being in the west, 19 being in the east. So we can see that a large number of blue individuals in the west and a large number of pure red individuals in the east, and a central area of admixture. When we map those admixed individuals onto the map, they belong to these 4 populations here. The distance between these 2 populations, the 2 most furthest populations, is 1 kilometer. So we have this, like, we've tightened up the region where we can see admixture occurring, and it's approximately coincident with the lowest elevation levels of that cloud ridge, as you would predict if hybridization has been facilitated by isolated high elevation populations now having recently moved down and come into contact through the cloud forest patches themselves coalescing together. Now I'm showing that same genotype data in a principal component analysis. So all of these individuals correspond to non-admixed individuals, pure blue. These all correspond to individuals that can be assigned to the red population, if you like. And here are our individuals sampled from these admixed populations. And what we can see here is that despite the narrow geographic sampling, we have a huge amount of genomic variation within this region, which is consistent with divergent genotypes having recently come together and admixed and recombined, generating this. This is exactly the pattern we would expect from recent admixture. And we can further confirm that recent admixture by simulating expectations for hybrid individuals based on parental genotypes. And so here we have simulating where we'd expect individuals to be if they were F1s, if they were F1 back crosses, or F2s. And we can see several of our individuals are consistent with F1 and F2 back crosses, telling us that this is ongoing hybridization. These admixed individuals aren't some relic admixed population. We have it's active zone of secondary contact and admixture between individuals. Mm. So this is really cool in a lot of ways, because we are going to go back in and sample more species with this, but this is enough to really tie it down and tell us exactly what is happening here. And it's the model is completely inconsistent with the standard model, but consistent with this model predicting that as climate warms, cloud forest lower elevation limits are pushed down slope. So that tells us that during cooler interglacials, cloud forest will actually be very limited in its extent, being limited to the higher elevation regions of the peninsula. As climate warms, as it has done, taking us into the current interglacial, that cloud forest is pushed down slope, expanding the geographic limits of the cloud forest, and will remain like that until we enter the next interglacial cycle, where cloud forest at lower elevation limits will be pushed up. And this is a dynamic that will repeat depending upon will repeat and will be consistent with the temporal duration of the glacial and interglacial cycles that we've seen over the Pleistocene. Now, in many ways, this might tell us something about the ecology of individual species. Those species that are more tightly associated with the cloud forest are more likely to have been impacted by the more subtle oscillations at the early Pleistocene stages, such as Acales globuli penis, whereas the more extreme oscillations we've more recently seen may have started to impact other less other species with with a less constrained niche, but with these more extensive oscillations. They've more recently become impacted and became isolated during the interglacial periods. Now, what's kind of nice for us is that looking in the literature, there is actually a climate, someone used existing climate station data about, was it 30 years ago, 25 years ago, and looked at a 30-year sequence of climate station data and came up with the same prediction, if you like, that as climate warms, cloud forests should be pushed down slope. Now, whether this is something that's a model that's applicable to all cloud forests around the world really is something that's, you know, I can't speak for that, but it's quite plausible that it would be, I think. And I think we can see a sensible way to test that in different regions just by applying this kind of approach when there's a suitable topographic framework. Right, now, so one thing hopefully I've maybe demonstrated or convinced you about is, you know, how we can take this sort of hierarchical approach to really understand the drivers of speciation diversification and what structures biodiversity. But at the same time, while we're able to overcome some logistical constraints, it still involves a lot of sample handling. What I'm just going to talk about now is where we're heading in terms of trying to make that even more feasible by further reducing logistical constraints. So what we did was we took individual specimens and individually extracted their DNA and then got an individual sequence. I mean, everyone's probably heard of metabarcoding now. So now, you know, what you can do now is take your whole community, which might be thousands of individuals, do one DNA extract, generate sequence data from that, well, amplify a gene of interest and generate sequences that correspond to that community. So that's like a hugely powerful tool, but at the same time, there are known limitations with the data. And one of those limitations is just artifact and noise. So we know that this data tends to overestimate how many species there are, and we can't really work with the interest-specific variation because we know there's a lot of sequencing noise and artifact within that. And while there's been moves to, there's been huge advances in dealing with that, it hasn't really taken us as far as we needed to go. So now I'm going to present the work from Carmelo and Duhar from my group, who has led an initiative to deal with what I would say was the last outstanding problem, which was removing nuclear copies from this data. So it's mitochondrial genes that have been inserted into the nuclear genome and persist there and amplify and sequence, and some copies retain signatures that are consistent with them being, like, if you like, live mitochondrial copies. That's a terribly difficult thing to deal with, but in this paper, if you're interested, Carmelo's come up with a really nice set of procedures to filter those nuclear copies out. And what's it enabled us to do is to then, for any given metabarcode data set, be able to say that we know that we have approximately 5% of our copies are not true mitochondria, 5% of our sequences, and we know that we're probably missing 5% or 6% of the real mitochondrial sequences. So we can put limits on error, and which really helps us to have some faith in working at the haplotype level. And I'm just going to give you an example of how nicely that appears to be working for us. So we went back to this peninsula and sampled soils from the same location, not the same locations, sometimes the same locations, but more locations, but from the same two regions. We extracted arthropods from those soils. Primarily you're dealing with collembola and mites, but there's also a large number of beetle species that are soil associated. So we extracted everything from there, did a single DNA extraction of all the extracted arthropods from the soil. It's about 20 liters of soil, and then amplified the CO1 gene and generated the metabarcode data, and then applied Carmelo's protocol. So what I'm showing you here is the original mitochondrial patterns we found for two species that can also appear in our metabarcode data. And here is the equivalent haplotype networks generated from the metabarcode data from those same two species. So you can see the same regional structuring of mitochondrial variation. So this tells us that we can now use this approach to generate broad community level patterns of phylogeographic structuring. And you know, I can't tell you how many times faster than compared to the way that we have been doing it. And it's quite reassuring that even when we calculate indices of population genetic structuring, they're fairly consistent between these two data sets. So this is something that's in, you know, we haven't published this yet. Camilla's in the process of putting it together now, but we think it's going to be a pretty valuable tool. What we have been doing is applying this tool to broader samplings across the island of Tenerife, with the idea of getting a handle on what we can call the dark texts, texts that we normally can't work with because they're just so difficult. Things like collembola and mites, where there's tremendous amounts of cryptic speciation, morphologically very hard to work with because of their small size, and very hard to work with because of their really high local abundances. So we extract the material from the soil. We do a single DNA extraction of everything. So it could be potentially thousands of individuals, generate the mitochondrial data, and then apply the filtering pipelines of Carmelo to eventually arrive at individual level species units with geographically referenced mitochondrial haplotype variation. Now, just doing this for 52 sites across the island of Tenerife, using a threshold of 3% to define what's probably a species. Per habitat, we sample between 278 and 316 species. And even when we apply a super conservative threshold saying, well, you know, everything below a similarity of 15%, we'll just call that a species. Even then we're finding numbers of species that are way higher than what we would predict based on existing inventory data for these groups. So for example, in Tenerife, there are recorded 287 species of mite, and applying our 3% threshold, which is actually not a bad approximation for where we should expect species to be, we recovered 434 inferred species. So 287 known, we covered 434. And you have to remember this is from 52 square meters of soil within an island that is 2,500 square kilometers. So we know that there is a huge amount of diversity present. We have and it gives us a powerful tool to understand how this diversity is structured by habitat. And we find very strong signatures for habitat structuring, which you may not intuitively expect for something that's soil associated, but things clearly divide by main habitat types. And what's really useful is that we can finally start to look at what's happening below the species level within these groups. So we have a number of species that are restricted to single habitats, but many that are distributed across more than one habitat. And this haplotype variation that we're able to recover enables us to identify those species that are actually structured by habitat. So whether that's incipient ongoing speciation or true speciation to biological species, we don't know, but we know that habitat is driving divergence within the species. And this is another clear example of that here, which is in 3 habitats, and variation is clearly structured by those 3 habitats. And it can enable us to distinguish these species from those that are in multiple habitats, but not geographically structured by habitat. But additionally, with this data, we can actually look at how to what extent dispersal constraints impact these species by looking at the extent of isolation by distance within species distributed across the landscape. Okay, I'm just going to bring my presentation to a close there. I think I've used up more than my 40 minutes already. just a final slide to acknowledge some of the principal individuals involved with this work. There's actually, you know, a lot more people have participated in the different projects that contribute to the talk, but these are people that have perhaps had the biggest input. Okay, and yeah, thank you very much for your time.